Welcome to this explainer. Okay, we're going to just dive straight in today because we are embarking on a really fascinating biochemical journey. We're going to look inside one of the most absolutely critical manufacturing plants in the entire human body, and that's the cellular factory responsible for making heme. Now, this isn't just some simple point A to point B process. No, it's a highly coordinated operation. It crosses cellular borders, and it actually solves a major biological mystery along the way. So if you love seeing how tiny, intricate cellular mechanisms all align to keep us alive, well, let's break this down.
To kick things off, we really need to look at a core biological paradox. We all know that mature red blood cells are just packed, literally stuffed with hemoglobin, right? And the essential iron-holding core of hemoglobin is heme. But here's the catch. Building heme requires some incredibly heavy cellular machinery. Specifically, it needs mitochondria. Yet, a mature red blood cell doesn't even have mitochondria. So how on earth is it mass-producing a molecule when it completely lacks the factory floor needed to build it? Well, the beautifully simple answer? They don't. The mature red blood cell isn't making heme at all. All of that heavy lifting is done long before the cell ever matures. The work is actually handled by precursor cells deep inside your bone marrow. You see, those early-stage cells have all the mitochondrial machinery they need to get the job done. Now, what makes this process so cool is the spatial map of our cellular manufacturing plant. Heme synthesis isn't something that just sits in one corner of the cell. It's a strict round-trip journey. The process starts inside the mitochondria. It moves out into the cellular fluid, the cytosol, for assembly, and then it literally has to return right back into the mitochondria for the final finishing touches. It's this beautiful, intricate dance across cellular compartments.
So, let's get into phase 1, the heavy machinery room. Imagine we're moving deep inside the inner matrix of the mitochondria. This is where the absolute very first step of our vital heme production takes place, safely isolated within those mitochondrial walls. To kick things off, we need a specific catalyst, an enzyme called ALS synthase or ALS. And honestly, this perfectly illustrates just how specialized our bodies are, because we actually have two different workers for this exact job. ALS1 is kind of like your general contractor. It operates in the liver and most other tissues in the body. But ALS2, that's our highly specialized worker, acting almost exclusively in erythroid cells, so the red blood cell lineage.
Okay, so what happens when ALS goes to work? It takes two chemical ingredients, glycine and succinyl-CoA, and it combines them to form our very first intermediate product, which we call ALA, or 5-aminolevulinic acid. Now, I want you to focus on that second ingredient for a second, succinyl-CoA. This is a massive detail. Succinyl-CoA is an intermediate from the TCA cycle, which is the main energy-producing engine of the cell. So by using this specific ingredient, the cell is brilliantly and directly linking the production of heme to its core energy metabolism right from step 1. It's just incredible.
All right, moving on to phase 2, the assembly line floor.
Once that ALA molecule is forged in the heavy machinery room, it doesn't just hang around. The newly formed ALA exits the mitochondria and heads out into the cytosol, which is basically the wide-open assembly line floor of the cell to continue manufacturing. Out here, the basic backbone of the porphyrin ring is built out through a series of really crucial checkpoints. First, two ALA molecules are combined by an enzyme called ALA dehydratase to form porphobilinogen, or PBG. Next, 4 of these PBG molecules get linked together to create hydroxymethylbilane. The molecule just keeps transforming, reshaping into uroporphyrinogen 3 and finally coproporphyrinogen 3. And throughout this massive cytosolic build, you've got these essential deamination and decarboxylation reactions taking place, basically shedding ammonia and carbon dioxide as byproducts while the molecular ring takes its final shape.
Now let's pause right here because I really need to warn you about a super common textbook misconception. Often, you'll read a summary somewhere that says, the initial steps of heme synthesis take place in the mitochondria while the subsequent reactions occur in the cytosol. If you just read that, you'd naturally assume the rest of the synthesis stays out in the cytosol until the whole shebang is finished, right? Well, that is entirely wrong.
That brings us to phase 3, back to the heavy machinery.
You see, phase 3 actually takes us right back to where we started. That coproporphyrinogen 3 we just built out on the assembly line floor, it has to dip back into the mitochondria. Once it's inside, it transforms into protoporphyrinogen 9 and then into protoporphyrin 9. The factory line has officially returned to the heavy machinery room, and the reason it has to come back is waiting right here on the inner mitochondrial membrane. Meet ferrochelase. This is the final, completely essential worker in our factory. You can basically think of it as the ultimate iron welder. Its one and only job is to take an iron ion, Fe2+, and insert it perfectly into the center of the ring we've spent all this time building.
And that brings us to the absolute most satisfying step of the whole process. Protoporphyrin IX meets that ferrous iron. Our ferrochelatase welder does its job, and boom! You have successfully synthesized heme. The round trip is complete. Mitochondria, out to the cytosol, and right back to the mitochondria.
But wait, how does this incredibly intricate, mitochondria-dependent factory process actually connect back to the mystery we started with? Remember, we asked how mature red blood cells carry so much heme when they don't have mitochondria. Well, it comes down to what we can call the precursor's sacrifice.
Let's map out exactly how this plays out. Back in your bone marrow, the red blood cell precursors, we're talking about erythroblasts and reticulocytes, are completely packed with mitochondria. During this early stage, they are running the heme factory at maximum capacity. They mass-produce heme, they assemble it into hemoglobin. But then, as they mature, they do something honestly incredible. They purposefully destroy their own mitochondria through a process called mitophagy. They literally eject the factory equipment entirely. So by the time it becomes a mature erythrocyte, the cell isn't a factory anymore. It's just a highly specialized delivery truck, carrying the cargo it manufactured back in its youth.
Now, as we all know, any good factory needs a quality control system, right? What happens if this intricate assembly line breaks down? Like, what if there just isn't enough iron available for our welder, ferrochelatase, to do its job? Well, the cell has a really ingenious emergency stop button.
Here is the big challenge. Red blood cell precursors have to perfectly match their heme production with their globin protein production in order to assemble hemoglobin. If heme synthesis fails, the cell can't just keep mindlessly churning out globin proteins that have absolutely nowhere to go. So how on earth do they coordinate this delicate balancing act across the whole cell?
Let's break down this biological domino effect because it is super clever. If there is a heme deficiency inside the mitochondria, it instantly triggers a stress signal. This stress activates a protein called OMA1. OMA1 then cleaves another protein called DLE1. Now, this cleaved DLE1 acts as a messenger. It leaves the mitochondria and moves all the way out into the cytosol. Once it's out there, it activates HRI. HRI then phosphorylates a key factor called EIF2 alpha. And that phosphorylation, that is the final brake pedal. It completely inhibits global protein translation, sharply reducing globin production. So it's this incredibly intricate multi-step biological alarm system linking the mitochondria directly to the cytosolic machinery.
Honestly, if we were to translate that complex OMA1, DELE1, HRI pathway into a simple factory rule, it would be exactly this. Don't make globin if there's no heme. The mitochondria act as the ultimate quality control managers, sensing a shortage and shouting out to the rest of the cell to just halt production. So, we've seen how intricately connected the TCA cycle, iron metabolism, and mitochondrial quality control all are to this one vital process. But I really want to leave you with this final provocative thought. Just think about the irony here for a second. A mature red blood cell is entirely defined by its ability to carry hemoglobin. Yet to successfully manufacture that vital hemoglobin, the cell relies utterly and completely on the mitochondria, an organelle that it must eventually destroy and discard in order to achieve its final mature form. It is just a breathtaking example of cellular sacrifice and biological complexity. I really hope this explainer gave you a whole new appreciation for the absolute marvel that is the Heme Factory.